Time to learn about hearing.
Hi, welcome to the lecture about hearing. This is the first lecture. Hearing as a theme will probably be cut in 3 or 4 different lectures. So this is the first one. Let's talk about sound. So what is sound? And this is a tough question. I mean, it's probably one of those questions that's pretty hard to answer from common sense. So what is sound? How does sound propagate through the air? What is it? If you think about that, you probably realize that sound is about vibration. Like you can think about a bell or a string vibrating, or maybe with a trumpet, it's like the air inside the trumpet that is vibrating. But there is some movement. I guess the easiest for me to imagine is one of those gongs that you hit in the middle and then they vibrate. And as this thing vibrates, it pushes the air, right? It moves a little bit, it pushes the molecules of the air, and then these molecules run into other molecules and they push them. And so what you have at the end is this wave, the wave of increased pressure and decreased pressure that propagates through the air. Every time this thing vibrates in one direction, it kind of creates a crowd of molecules, and then this crowd of molecules like hits other molecules, and you have a wave. But each time it goes in the opposite direction, it creates a rarefication of air, which is a funny word. And these areas of slightly higher pressure and slightly lower pressure propagate through the medium. That's sound. All right, so hearing is about detecting this. And hearing can also work underwater, right? So it doesn't have to be air, but we'll be talking about air mostly because just we're biased towards our ears, and our human ears work in the air. Okay, good. And also, if you plot, like if you make a graph of how pressure changes as you go through the air, you have this nice sine wave that you can see. So it goes up and down, up and down. So this is the graph that you have if you make a snapshot, like you freeze time and then measure pressure in different parts of the room. But you have exactly the same plot if you don't freeze time and place an ear in one place of the room, because these waves are propagating. So your detector, in this case the ear, will be hit with waves of higher pressure and waves with low pressure again and again. So the detected pressure will go up and down, up and down, up and down, and that forms the sound wave. So we now need to build a detector that would work with something like that would detect it, and that will eventually figure out what the heck is going on. Because we don't just detect the presence of the wave, the magic of hearing, as with any other sense, is to figure out what's happening in the world. So we need to do something fancier than just detect it. We need to be able to analyze that. And it's not a trivial task, as we will see. Okay, what are the 3 main parts of a human mammalian ear? That's not something that you can figure out on your own, but that's something you probably heard about one way or another. Think about it. What does the ear consist of? We have the outer ear, the middle ear, and the inner ear. What's inside the outer ear? Like, there is a canal called an auditory canal that leads towards the middle ear, that leads inside your head. What's inside this canal? What is it filled with? That's a stupid question, right? Of course it's filled with air, because the outer ear is open to everything that's outside, and it's air outside, so there has to be air in the auditory canal. Okay, good. Done. First question answered. Now, what's the border between the outer ear and the middle ear? Okay, let's see. A thing called the tympanic membrane. Another name for this thing is the eardrum. So that is a membrane that's stretched kind of across the auditory canal, and it separates the middle ear from the outer ear. So there is no direct passage of air. If it's ruptured, the person loses their hearing, so we don't want to rupture it. Okay, what's inside the middle ear? So we figured out that the outer ear is filled with air. What fills the middle ear? What's in here? What are the options? Okay, let's skip this question for a second. Let's look at the inner ear. The inner ear has this funny shape, and what's inside the inner ear? So the inner ear is where the detection happens, the actual detection of sound. If there is a detection of sound, then there have to be detector cells. Detector cells are alive, right? If they're alive, they have to be moist, right? Remember, cells, they have a membrane, they have to be intracellular fluid, but they have to be extracellular fluid as well. Cells cannot be exposed, like our human cells, cannot be just exposed to the elements. They will dry out and they will die. So in order for these detector cells to work, the inner ear has to be filled with fluid. So while the outer ear is filled with air, the inner ear is filled with some sort of fluid. It has like salty water inside because it has detector cells. Now the question is about the middle ear. It's kind of in between. So it's separated from the outside, but it's also separated from the inner ear. What is it filled with? Air or fluid? We have basically two options. What do you think? That's a hard question, right? You might remember that it's actually filled with air. It has air inside. There are two ways to remember that. One, we have this interesting passage. This passage below leads into our throat. So it opens inside our throat somewhere deep in there, which means that the inner ear is connected with atmosphere just really indirectly. There is a thin passage that goes from the ear into the throat, and then throat, because we open our mouth every now and then, is connected to the atmosphere outside, which means that inner ear is both protected from the elements, but at the same time the pressure inside should be the same as the pressure in the atmosphere, which is actually important if you ever drove a car up into the mountain or like rode an elevator or something like that. Or if you ever descended from a mountain fairly fast, you know that something happens with the ears. Same happens actually if you dive underwater. If you quickly change how deep you are or how high you are above the sea level, the pressure changes outside. And if your middle ear cannot equalize pressure, you feel this pain. And as it equalizes pressure, you actually hear that because your ears pop, I guess is the word that's usually used. And also if you have a cold or if your nose doesn't quite work well, so if everything is slightly swollen, then this tube that is supposed to lead to your throat, this one, it may get swollen, in which case it won't equalize and you may have to blow your ears, like do this, you know. If you've ever done that, then you know how it works. And that's a reminder that the middle ear is filled with air. Another reminder, kind of another way to connect, if you ever had an ear infection, you might have heard that your middle ear can be filled with fluid and then you cannot hear well. And this fluid needs to be drained at some point. If it doesn't drain through this tube, it may have to be drained medically, I guess. Okay, now let's talk about the outer ear first separately, and then about middle ear, and then about the inner ear. Why outer ear even exists? I mean, why do we have these things, you know? Why do we have them? Why wouldn't we just have an opening and be done with it? What do you think? That's a simple question, you can answer it. It's to collect sound, right? Look at this. Look at this animal. Do you know this animal? This is a pup of a fennec fox. It's like pups are cute to begin with, but then fennec foxes are also cute, so this is like cute square. But fennec foxes have these giant ears, and they have it because they hear really well, but also they live in the desert, so they use them to cool themselves. But anyways, it kind of shows how big the ears can be, and they collect, among other things, they collect sound. So all the sound waves, all these changes in pressure, when you collect them over a bigger surface and kind of channel them into a smaller opening, you collect the energy of the sound wave and you make the sound wave louder. And those old school gramophones, like this one on the left, they kind of work in a similar way, just in the opposite direction. So you have relatively powerful but, you know, small in area movements of the needle, and you extend them over a wider area to produce more sound. So in either way, it's about redistributing energy. In a gramophone, you distribute the energy of the movement of the needle and turn it into sound. In this case, in case of our ears, you catch the sound and make it move something with more power. And in case of our ears, what does the sound move eventually? The tympanic membrane, the eardrum, right? We'll get to that. But so yeah, basically the main reason we have the pinnae, that's how these things are called, is because we collect sound. And people use that during the First World War and sometime after that, before radars were invented. So before people figured out how to use radio waves to locate airplanes, the only way to locate an airplane when the sky is overcast, if it was flying reasonably high, like in the clouds, the only way to find it, to identify it, was to listen to it. And people use these essentially extensions of their outer ears in order to locate airplanes. So in this case, on the right side of this picture, you have this giant setup, and somewhere in the middle over here is a seat, and the person would be sitting there. So you would climb some sort of ladder, sit there, and you would have some rotation knobs, so you would rotate it for this entire machine to point at a certain part of the sky, and then you would take two tubes and plug them into your ears in order to listen to the sky. And armies had these machines always like on, in the sense that there was always a soldier sitting inside listening to the sky to warn everyone about approaching airplanes. Here's another picture of a kind of intermediate size machine that shows how it looked like. Okay, next, why do we have a middle ear? So this is kind of a hard question, but also one that you could potentially answer using common sense, although it is hard. So I will ask you to pause the video and think about it for a while and then compare your answer to my answer. Why this question is important? We have outer ear. We have to have some sort of outer ear, right? Because one way or another we have an entrance from the atmosphere into our head. So the outer ear is a given. The inner ear filled with fluid is also a given, because you have to have detector cells, whatever they are. We will talk pretty soon what they actually are and how they look like. But there have to be some detector cells, and these detector cells have to be in the fluid in order to function, because all cells need to be surrounded by fluid. And also you probably can guess that detector cells have something to do, probably do have something to do with voltage, because so far all detector cells we discussed had something to do with membrane voltage, right? They were either neurons or almost neurons, like neuron compatible cells, so to say, which means that there will probably be some ions in the outer fluid and some ions in the inner fluid and some interaction of those. So they have to be surrounded with something in order to function. The inner ear has to be filled with fluid, and the outer ear in humans and in birds, in dogs, in crocodiles, in everything that lives above ground has to be filled with water. So these two parts have to exist. But middle ear, why do we have it? This is less clear, right? Why wouldn't you connect the outer ear to the inner ear directly? Why do we have this additional part in between? And the part that has some machinery in it, because you've seen on the previous picture there is a tympanic membrane, but then there is something. That's how this something looks like. There are some tiny bones called ossicles. Why? Pause the video, think about that, then compare your answer to the answer in this video. Done? Okay, let's talk about that. So here's what happens in the middle ear. We have this auditory canal, right, that leads from the pinna inside your skull. It's filled with air. The pinna catches more sound, channels it into the canal, but it's still sound. So there are still waves in the air, still areas of higher pressure and lower pressure going through the air and hitting your tympanic membrane and moving it. Your tympanic membrane moves, and we need to somehow send this motion into the fluid of your inner ear, because you have cells that have to be alive, so they are surrounded with water. How to make it happen? One potential option could be this naive approach. Let's just put the tympanic membrane directly between the outer ear and the inner ear. There is a problem with that though. And the problem is that water or fluid, salty water, fluid, but essentially mostly from the physical point of view, it's mostly water. Water is pretty heavy and it's not compressible. What does it mean that it's not compressible? Like with air, you probably know you can compress it. if you ever biked, if you ever had a bicycle and you tried to use a pump, an air pump, you know that you can compress the air and you release it, it expands back. Imagine a balloon. A balloon would be probably the best example, an air balloon. You can kind of compress it a little bit and then it relaxes back. So it doesn't work with water. You cannot compress water. If you ever played with water soakers, that's how soakers work. If you push water in one part of the soaker, it immediately creates a stream on the other part. Water is not compressible. Why is it relevant here? Because if you just make the air shake the membrane, and if there is water behind the membrane, it will be pretty hard to transmit sound into the water. Water will resist that. It's heavy and not compressible. So in this case, the membrane will kind of contain the water, but essentially the result will be about the same as if you just shrieked at the surface of the water. So if you imagine like a cup of water and you're speaking to it, will it be possible to hear you if the microphone is in water? Now, if you ever tried to dive in a swimming pool, you know how sounds of the outer world from the air, how can they be heard when you're in the swimming pool? You can hear a little bit, but not much. Basically, all higher frequencies, like all these sorts of sounds, all the whistling, noisy sounds that are important, for example, for understanding speech, they all get reflected from the surface of the water. The only thing you hear if you're diving are the lower frequencies. Like it sounds approximately like, right? That sound familiar? If you ever tried to dive underwater, you might remember that, because the water reflects most of the sound. So it's not a good idea to just have a membrane separating the fluid from the air. This ear won't work that well. You can hear low frequencies, but you won't be able to hear high frequencies. And high frequencies in our life are usually more informative. All meaningful noises, all noises made by small objects, produce more high frequencies than lower frequencies. So we have to find a way to make the air shake the water efficiently. We need some sort of machine, almost an amplifier that would make it happen. And that's exactly what the middle ear is, because it has these ossicles. And we humans have 3 ossicles. And these ossicles can articulate with each other. That's how they call it, which means that they have something like joints and they can move relative to each other. So it's not one mass, it's like a machine, a system of levers. And what happens is that they take the vibration of the tympanic membrane and they transmit it into a much smaller window, which is called the oval window. It's over here. And they shake the water inside the inner ear. So it transduces vibration and it also amplifies the vibration from the air and into the fluid. Now, this probably sounds a little bit abstract to you, like, okay, some little bones that can move relative to each other and somehow they amplify the vibrations. Like, how does it work? Can we relate to that? Okay, let me show you the simplest approach. The simplest approach to amplifying sound. Imagine that you have the outer ear. I will be drawing. So imagine that you have the outer ear. This is the canal, auditory canal. So this part is filled with air and you want to transmit it into the fluid. So there will be some other area that's filled with fluid. One way to amplify it is to put something here that would catch the vibrations, which is called the tympanic membrane, and then connect it with a much smaller piston-like thingy that would shake the fluid in the inner ear. Like imagine something like that. If you think about it, as this part moves, this part will also move, but the force collected will be kind of pointed to a much smaller area, and that will make the pressure there higher. Like this also sounds abstract and physicky, but think about it. For example, if you lie on the floor and your friend stands on you with their feet, that's wild, but it can happen, who knows, maybe you're dancing some contemporary dance. You will survive that, right? A person can stand on you, like not on the face probably, but someplace on the back if they're careful. You can totally handle their weight, right? But if they put some sort of a pointy triangle on you and stand on it, so don't do that. This is a leg, because then this entire weight will be concentrated in this single point and it will be driven into whatever is under it with too much force, right? That's how knives work. You concentrate the entire force of the hand on a tiny point and it goes into the butter or into the broccoli, whatever you're cutting. Right, so something like that happening here would also work as a way of shaking the fluid. You collect the pressure over a wider area, and then by making this ossicle connected to a tinier piston, you concentrate it over a smaller area, and so the force is greater, and you can actually shake the fluid. That's a one ossicle ear, but it's kind of like our ear, in the sense that we also have the wider area, which is called the tympanic membrane, and we have this smaller part, which is called the stapes, and it's connected to the oval window, and it shakes the fluid inside the inner ear. That works. We can actually, even with one ossicle, we can make it slightly better. Let me show you how to make it better. So let's do the following. Let's take this membrane and connect it to a long piece of equipment that has a hinge here. So it has a hinge, so it can rotate like that and like that. And then over here below, we'll add the same piston that we had before. But now it's not exactly against the membrane, but it's placed on a lever. What will happen in this case? We'll push the fluid inside the inner ear with even more force, because this thing will act as a lever. So larger movement of the tympanic membrane will translate into a proportionally smaller movement of this part, but it will have more force to it. It's kind of like those nut crackers, those devices you use to crack nuts, you know? Here, I mean something like that. Bigger give of the arms, smaller give inside the part that actually breaks the nut, and you have greater force. So your ear kind of works like that, except here I drove a one ossicle ear, and many animals have a one ossicle ear, frogs for example. Reptiles, crocodiles and lizards have one ossicle. Birds actually also have one ossicle. We have three, but it doesn't change much. It just makes it possible to condense it kind of into a smaller total volume, because you don't have to have this long lever. So it's a 3-lever system instead of a 1-lever system, but it serves the same purpose. Now you know why ossicles are there. In order to transmit the vibration in the air into the vibrations in the fluid, and also amplify them to a degree. Now here's an interesting question, which is an offside. It's like a footnote, but I think a pretty interesting footnote. What do you think? How can we make these ossicles light? Like by we, I mean, imagine that you have to reverse engineer this ear. So again, you're Archangel Gabriel, and the Lord asked you to make a nice working middle ear for those pesky animals that walk on the ground, because you have to transmit vibrations from the air into vibrations in the fluid. So you came up with this plan that you will have a lever and it will be rigid. And what are the rigid materials that you have in the body? Bones, of course. Okay, so it will be made of bones, because that's the only way for us to build something of an intricate shape that would be rigid. Good, it have to be built of bones. But now to work, it has to be fairly light, and it's surrounded with air. And if you think of it, that presents a problem, because do we have any other bones that are surrounded with air? No. All of our bones are nicely covered with flesh, because bones also needs to be maintained, right? So there are also live cells. In some bones, we also have bone marrow, but these bones are probably too small, but still, there are some cells that build bones, that repair bones. So if there are cells, they have to be fluid. So these bones should be submerged in the fluid. But these guys we just discussed are by definition in the air. And also they have to be light, but real bones, like live bones, should have some blood supply, right? Another reason, if you want cells to be alive, they have to have oxygen, they have to have nutrients. So something needs to bring these nutrients, so there have to be vessels, blood vessels. But you want this thing to be lightweight and transmit vibrations all the time, so can you afford vessels? And it's in the air anyways. How does it work? How can we make these ossicles, these tiny bones, out of bone and make them work in the air at the same time? There is a conflict here. What's the answer? What's the solution? Think of this question, pause the video, think of it, then continue the video and compare your answer to mine. What do you think? This is a little bit of a trick question. Maybe figure that out. Bones have to be alive, but these bones have to be in the air. And no live bone can be in the air, at least not easily. What does it mean? That these bones are dead. Yeah, you heard it right. You have dead bones in your ear. They are indeed surrounded with fluid. When we are embryos in the womb, it is required for them to be built, for them to develop, for them to be constructed by these little cells that make bones happen. But then once they're done, the fluid is drained after we're born, and now they're in the air and they die. I find it kind of interesting, and it sounds really nice. In order for you to live, something inside you had to die. That's cool, isn't it? Okay, let's go further. How do these bones articulate? They cannot be just fused to the skull, right? They are hanging on ligaments. By the way, what is this stuff around? This is the skull, right? So all of that happens in a little cave inside your skull. And these bones are hanging on ligaments. This is what makes hinges. And some of these ligaments are also connected to muscles. These are tiny, tiny muscles. There are two of them. And they do funny things. They do dynamic range adjustments. And another thing that I loosely call de-chewing, although I'm not quite sure how it's officially called. What is the dynamic range adjustment? Well, if you ever try to talk to a human while on a subway or in any other loud place, like in an airport, in some place where there is noise everywhere, and you talk to a human and they're practically shouting in your ear, right? Have you ridden a subway train ever? So they're like close to your ear and they're like, yeah, and then I went there and it was like you wouldn't believe. They're screaming in your ear and it's kind of unpleasant, but it's tolerable and you can function. Now, if you were sitting in the library in the quietness and somebody came to you from behind and suddenly screamed in your ear with same intensity, they could very well rupture your eardrum. At the very least, it would be very painful. You can imagine that. A sudden loud noise, if you're sitting in a quiet place, could damage your hearing, like for real. Don't do that. Don't scream into each other's ears unexpectedly. It's not healthy. Why is something that's tolerable in a loud setting will totally destroy your ear in a quiet setting? Because we have a system in the ear to change the sensitivity of our ear to the sounds. And this system is entirely mechanical, which is pretty cool, right? There is a muscle that goes to the tympanic membrane. And if this muscle relaxes, the tympanic membrane can move freely. And then we can catch tiniest sounds, and that's how you function if you're walking at night in your house, or if you're trying to eavesdrop on a traitor who is trying to kill the king, or if you're in the forest and you're trying to catch the fox. In all these situations, you will be listening to quiet sounds, and this muscle is called tensor tympani muscle. It will be relaxed. Your tympanic membrane will be able to move and respond to sound as much as it can. If you're in a loud setting, for example, knights are riding to the battle and there is armor everywhere and it's clanking and horses are neighing. In this situation, because there's lots of sound, it would be nice to lower the sensitivity of your ear. And indeed that would happen. The tensor tympani muscle would stretch your tympanic membrane, and stretched, it won't be able to move that freely. It will be tense and it will move much less, which means that you cannot hear tiny sounds, but you wouldn't be able to hear them anyways because there is so much noise, but it would move less in response to loud sounds, and it would not damage all this machinery and your inner ear as well. So that's one thing, dynamic range, and that's very important and directly related to safety of hearing. The same sound can be safe or unsafe depending on whether you're prepared to hear it. But then another funny thing is what I call roughly de-chewing. And here's what it is. Our ears are inside our heads, right? They're sitting inside our skull. Now, sound propagates much better through the solid medium than through the air. If you remember those pictures when people listen to horses far away by listening to the ground, it's actually kind of doable. Sound propagates better in solid objects than it does in the air. And we have our sensory organs for sound inside the skull, but also inside the same skull, we have the chewing part, we have all the teeth. Imagine how much sound we make when we eat, because we use the same skull that harbors the ears in order to crush food, which means that our eating is actually incredibly loud in our head, and yet we don't hear it. Other people eating seem to make more noise compared to us eating, even though we have the listening thingy as close to the crunching thingy as it gets. How is it possible? Partially, it's probably filtered, certainly. It's certainly filtered in our head, so our brain somehow expects the sound that will come and tries to ignore it and does a pretty good job ignoring it. But partially, that's mechanic. And it works in the following way. When we eat, every time we clench our jaws while eating, our ear uncouples. I'm not sure which muscle is it. Is it stapedius muscle or tensor tympani, or both of them, but our ear, this transduction mechanism in the middle ear, is turned off. So we go temporarily deaf, or at least somewhat deaf, every time we bring our teeth together. And there are three ways to illustrate it. One way is to eat in a loud place, like not particularly loud, but like middle loud. For example, a place with steady noise. You can probably simulate it with your headphones as well. If you turn on some white noise, or if you're eating, say, at a train station. As you eat, every time you chew, you'll hear the background noise kind of disappear transiently for a fraction of a second, because you would go deaf temporarily. At least for me, it doesn't work if I just clench my teeth if I'm not eating. I'm not sure how it works with other people. You know, I kind of learned while teaching this course that people are slightly different. So it's quite often that one person has it and another person doesn't. Like, remember, the photic sneeze reflex and so many tiny neurological things are actually different in different people. But at least for me, if I eat in a loud place, I hear the noise dropping, and I read in the book that it seems to be a common thing among people. So that's one way. Another way to appreciate just how loud it is to have somebody else eat for you. So here's how it works. If you have a friend, do you have a friend? If you have a friend whom you can like touch with your head, like it's kind of hard now because of COVID, but if you have a relative, I don't know, a significant other. So this is you, okay? This is your head. I'm sorry. This is you. You have tiny eyes. So you won't be eating. You will be listening. You have these ears. Make another person touch your head with their head. So just bring your heads together. You don't have to do it now, but at some point, it's fun. Especially if you have something like cornflakes, something that's really, really crunchy. So eat some cornflakes yourself, listen to how it sounds. Well, loud, but not particularly loud. Then give them some cornflakes and make your heads touch. So this person, they will be crunching cornflakes, okay? I'm not sure how to draw it first. Like this, no, that was bad. Anyways, they are eating cornflakes. They will produce the crunching thing. They won't hear it themselves that much, but it will shake their skull. Their skull will shake your skull, and you'll listen to them eating cornflakes approximately as loudly as they would have heard themselves eating cornflakes if this sound weren't filtered out by the inner ear. Does make sense? And it's unbelievably loud. It's unbelievably loud. You wouldn't believe me. Just try that. Now, if you don't have friends right now because of the pandemic, if you don't have friends, there is kind of one more way that I will try to demonstrate now, which involves transmitting the sound in the outside and then listening to it coming from the air. Let me try to show how it works. So here how that works. Take something crunchy and then also take a guitar or an ukulele or something. Put something crunchy in your mouth, then put your mouth, let it touch the guitar, and then eat the crunchy thing. And in this case, the vibration of your skull, of your joint in this case, will go directly into the wood, and this wood is made to be loud because it will move, it will resonate, and it will project the sound into the room. Here's how it looks like. Right? That's ridiculous. That's ridiculously loud. So every time you chew, your nice ear dampens that. Apparently, in some people it doesn't work, and their own chewing annoys people to no end because it's so loud. Next, now it's time to talk about inner ear. And the inner ear has this cochlea thing, and the cochlea thing has this funny shape. Here's the picture of the entire system again, and the cochlea is this spiral part that kind of looks like a snail. So one thing to keep in mind is that it looks like a snail, sure, but you actually cannot cut it out from an animal or a human. You cannot cut it out and look at it like that, even though that's how it's shown in all the textbooks. Why? What is it surrounded with? It's surrounded with bone, right? This thing is actually a negative shape of sorts. That's a shape of a hole, a cave inside the bone. It's not an object that's surrounded by softer tissue. It's an empty space inside a harder tissue. But that's the shape of this empty space. It has this funny shape. It looks like a snail. Why does it have this shape? Why is it spiral? That's one question. This is the simpler one, a simpler one. A harder one is why do we have cochlea in general? So let's start with the spiral one. Why is it shaped as a spiral? To save space, right? That's easy. It's shaped like a spiral because it actually wants to be long. In some species it is long. In birds, for example, it's pretty much like a long thing that just slightly curves to the end. But in mammals, it's curved like that, which means that a long structure is condensed into this little space. But why do we have cochlea? What's the point of having cochlea? Now, this is a tough question, so I don't expect you to know the answer, because it relies on a very peculiar statement that's actually not even biology and not even physics, but rather math. I will state it very loosely now, but then later we'll talk a little bit more about that. Here's the statement. Sound can be represented as a bunch of sine waves, a bunch of these nice oscillations of different frequencies superimposed together. So if you have a sound, it's like you have this frequency, and then this frequency, and then this frequency. So sine waves of different frequencies and different amplitudes kind of thrown in together, summed up into one waveform. It's kind of hard to believe in it, but let's just believe. Let's believe that if you have a sound, it can be disassembled into these elementary frequencies, and that if you have elementary frequencies, you can resynthesize the sound. We will talk a little bit more about that in the next part of this hearing section, but a very simplistic explanation. Sound is changing the pressure of the air. Sound is always about vibrations. And because it's a vibration, it never deviates from the base atmospheric pressure too much. So it never the pressure never drops to zero. It never grows into infinity. It just slightly more, slightly less, more and less, more and less, moving around this basic pressure, which kind of suggests that maybe if you have a bunch of sine waves that also oscillate around the central pressure and you just mix them in just the right way, pick which ones to mix and how much of them to add, maybe you can resynthesize various sounds. And if the sound changes in time, like the snare drum, you'll just add more frequencies of some type in the beginning, and then you will change the volume of them. So maybe more bass in the beginning, more high frequencies at the end, something like that. So let's just believe for now. Let's just believe that every sound can be disassembled into elementary oscillations and can be assembled back into elementary oscillations if necessary. How does it help? It helps because that's how cochlea works. But before we go into that, stop this video. Look in the description. There is a link to a different video, not mine, called Auditory Transduction. That's the best video about the whole process of auditory transduction ever, and it's about good time now to watch it, not once but 2 or 3 times. Watch it several times because it has a 3D model of the inner ear and of the middle ear as well, and it shows how all these things move relative to each other and how they arrange relative to each other and how they function, and it explains how the cochlea functions. Watch it, then return back to this video and continue from this point, okay? I'll be waiting for you. Are you back? Good. Cochlea analyzes frequencies. Different parts of the cochlea are sensitive to different frequencies. That's wild. I mean, why, right? Why would you use something like that? You receive this sound. Here how it looks like. This is the time axis. Time goes like that. This is pressure. Pressure goes like that. You have the signal. Why wouldn't you encode the very signal? Why wouldn't you encode the signal in spiking one way or another and send it to the brain? It turns out it doesn't work like that. Why doesn't it work like that? Because let's think of that. The action potential, this thing, one spike, is about 1 millisecond long. Like the up phase, this part, the actual spike is 1 millisecond long. Together with the refractory period, it's maybe 5 milliseconds long. So let's go with 5. If it's 5 milliseconds long, 5,000ths of a second, what is the highest frequency that you can have? Highest frequency of spiking. At the best possible case, if the neuron is doing its absolute best, it would generate an action potential every 5 milliseconds, which means that you will have 200 spikes a second. And that's 200 hertz. All right, now 200 hertz is actually a sound. Yeah, we can hear it, absolutely. But is it a high sound or is it a low sound? It's pretty low. Let me play a 200 hertz sound for you. That's approximately it. This is approximately 200 hertz. Like if it were one frequency, it's actually more than one frequency, we'll talk about that. So this sound you can encode with spikes directly. And you could, like everything lower, you would also be able to encode. But everything above, you kind of cannot, like that, but also that, but also that, but also like any noise-like sounds, because their frequencies are even higher. You wouldn't be able to encode them. And most real sounds are pretty high frequency. So even though a snare drum has this nice shape, you just wouldn't be able to catch up if you tried neurons to spike every time the pressure goes up and not spike when pressure goes down. It feels like a reasonable way to encode sound, but it just doesn't work because the process that we use in order to think is way too slow to catch up with the changes in pressure in any reasonable sound. So instead, we have to rely on these very different and mathematically weird process. We disassemble sound into separate frequencies. Different parts of the cochlea are sensitive to different frequencies, and they shake differently. For higher frequencies, like woo, what part of the cochlea shakes? Closer to the entrance or further from the entrance? Its parts closer to the entrance. What is the entrance here? The entrance is that part that is shaken by the stapes. It's called an oval window. So the tympanic membrane through the ossicles shake the stapes. The stapes moves, makes the oval window shake, and the sound, the vibration enters the cochlea. And now the cochlea is arranged in such a way that parts that are closer to the entrance are shaking with higher frequencies, while parts that are further from the entrance are shaking with lower frequencies. How does that work? So two parts to this question. First, why would one part shake with one frequency and one part with another? It's pretty hard to imagine. But also the second question is how to make the cochlea tuned, different parts of the cochlea tuned to different frequencies. Let's start with the second one. Let's think, if you have two similar objects of different length, one of them is longer or wider or larger, and one of them is smaller or thinner or narrower, which one will make a lower sound and which one will make a higher sound? A bigger object or a smaller object? Think about that. Like a guitar is a good example. If you have a full string, it makes a sound. If you make the string shorter, it makes sounds that are higher. So we can partially tune the cochlea by just making different parts of it, different parts of whatever vibrates in it, have different size. In this case, it's different width. So what happens is there is a membrane called the basilar membrane that separates one part, one channel in the cochlea from another channel. Here's what happens. This part, the oval window, is connected to the stapes. The stapes comes here. The stapes moves, which means that the fluid here moves as well. The fluid here moves, and then the fluid here also moves, and then the fluid over here also moves, right? But all of this thing runs along a basilar membrane that separates this canal from another canal. And they're called scalars tympani and scalar vestibule, but unless you're preparing for MCAT, the names don't mean that much. Just know that there is like one tube and another tube, and they're separated with a flexible membrane. So because they're separated with a flexible membrane, if there is a motion here of the fluid, it doesn't actually have to go all the way to the apex, turn, and go all the way back. It doesn't have to. If it can cross through this flexible membrane, but the flexible membrane should have some give to it. It should start vibrating. If it starts vibrating, then the motion kind of crosses from this path, from this one to this one, from the one on the left to the one on the right, earlier. So it tries to cross over here, and then it tries to cross over here. It tries to cross in every part. And if it fails to cross, only then it crosses in the apex. The simplest way to draw the cochlea would be like that. You have the stapes here that shakes the oval window, and then the fluid moves. And if the vibration is really slow, then the best that the fluid can do is to just get displaced and cross there in the furthest point from the window, which is called the apex, and move the fluid over here. And there is a window here called the round window that has a membrane, and this membrane has some give to it, and so this membrane will kind of move to accommodate for this motion of fluid inside. But if it's not just a displacement, but more of a vibration, if it goes here and back, and this membrane that separates one scala from the other, one canal from the other, can also move left and right because it's flexible, then the vibration will be transmitted through this part, and there will be no motion in the apex. So now it's just the question of which part of the basilar membrane can vibrate at different frequencies. And as we just discussed, smaller objects tend to vibrate at higher frequencies. Thinner objects tend to vibrate at higher frequencies. So the beginning, close to the opening over here, the basilar membrane is narrower. This is in the corner a different picture of that. Closer to the beginning, it is more narrow. And further from the beginning, closer to the apex, it's wider. It's kind of weird because the canal kind of focuses in one direction, but the membrane focuses in the other direction. The canal becomes more narrow as you go further away from the entrance, but the membrane becomes wider as you go away from the entrance. That's a bit confusing, but concentrate on the membrane, all right? Another thing also changes about the membrane, it's stiffness. So let's think about stiffness. If you have two thingies that are similar in length and size, but one of them is more stiff, one of them is more tight, and another one is more relaxed, which one will vibrate at a higher frequency? Stiff or relaxed? Stiff, right? These are fairly easy questions in the sense that if you ever played with a piece of rubber, you know that as you stretch it, the frequency goes higher. Same in the guitar, all the strings have like the same length unless you press over here, but those that are wound tighter have higher frequencies than those that are more loose, right? And if you rotate these parts, you can change the frequency by just tightening the string or making it more relaxed. So same in the cochlea, because the basilar membrane closer to the entrance needs to be tuned to higher frequencies, it is more stiff, while basilar membrane further from the entrance, closer to the apex, is kind of more flappy and responds to lower frequencies. And so what happens now is, imagine that some sound comes that consists of some high frequencies, but also some low frequencies. So there is some ringing and whistling that's really high, but also some frequencies that are lower. A mixture of both frequencies will be fed into the system. It will move the tympanic membrane, it will move the ossicles, it will move the stapes and the oval window. And so it will move the fluid. But then higher frequencies will resonate with some part of the basilar membrane. So somewhere here in the cochlea, some part of the basilar membrane will be able to move back and forth on this frequency, and which means that this higher frequency will cross the basilar membrane earlier. It will make the fluid in the descending canal move earlier. The high frequency will cross here, and the movement of this fluid above this point won't have the higher frequency anymore, but it will still have the lower frequency. And the lower frequency will travel until the part that can vibrate in response to lower frequencies, and that's where this membrane will move, and it will make the fluid in the descending scale move. And the lowest frequencies that cannot be perceived by our ear won't make any part of the basilar membrane move, and they will reach the apex, so they will continue going and going like that, and they will reach the apex and they will just cross here. So it's equivalent to crossing like that on this scheme. This is wild in the sense that it's pretty hard to both understand why it happens like that and how it happens. So you may have to read about that in the textbook and then maybe rewatch the auditory transduction video and then maybe watch this video once again. And then also think of yourself shaking some membranes, like shaking a link fence or trying to open a door. There is a lighter, smaller door, and you can shake it at a higher frequency, and then there is a really heavy, large door, like a gate, that you can also open and close, but it's so large that you can only shake it at a lower frequency, something like that. You have to kind of find your mechanical intuitions in order to feel how this thing works. And even after you do that, it's still kind of wild, because the very idea that in order to understand sounds, we disassemble them into separate frequencies, then this information is actually detected, which is the next topic, how it is detected, and sent into the brain in this disassembled form, only for our brain to reverse, like to mix it back in some way, like not directly. We don't have like, you don't have real music playing in our head, but to understand what the sound was from this frequency-based picture, that's wild. But okay, let's go further. So now as different parts of the basilar membrane move at different frequencies, how can we detect that? Well, it turns out that there is a special thing that's sitting on this basilar membrane, which is called the organ of Corti. And this is a funny thing because at a cross section, it looks like a tiny thing. And here's a zoomed out version of it. But actually, this part is a cross section of the cochlea. So cochlea is this long thingy that's turned into a snail. And along this entire three-dimensional snail, we have sensory cells that also run in this spiral way, right? So it is a very long organ that has a structure represented by a cross section. It has special sensitive cells called hair cells. Here they are, and here's another one. And this is a picture from a normal textbook, like normal undergraduate level textbook, because actually all of these supporting cells are known. They are pretty stereotypical. They all have names. If you go to a doctor, to an otolaryngologist, the ear doctor, or if you look online, you can find detailed descriptions because each of these cells has a name. Most of them are named after some people who described them in the 19th century, I guess. All of that machinery is well described, but we will just concentrate on the main part, main player. And the main player are the hair cells, these ones and these ones. And the hair cells are curious. It's a cell, so as every cell, it has a cellular membrane and it has a nucleus and it has some stuff inside, but also it has a special thingy, a special organ. And it's not like hair. It has nothing to do with like the hair that grows on our heads. Nothing. So it's not whiskers, it's not sensory hairs on a manatee. It's just the name. The name is the same, the cells are completely different. There are sensory hairs on Drosophila, there are sensory hairs on a mouse. This is a different story. Like, don't mix them up, please. Hair cells are called hair cells just because if you look at them under a microscope, they have this thing on top. And somebody needed to give them a name. All right? That's an important point. It's like trivial and naive, but important. Okay, hair cells. What do? It turns out that they can feel the displacement of these protrusions. So they have some protrusions. And when these protrusions go in one direction, cells change their voltage. And when they go into other direction, they change their voltage in the opposite direction. So when they go in one direction, they depolarize, the voltage increases. When they go in the opposite direction, they hyperpolarize and the voltage decreases. They change the voltage of their membrane depending on the position of these protrusions, of these stereocilia, and the way they are placed on the basilar membrane, this basement on which they're sitting is the basilar membrane. The way they are placed is that when the basilar membrane shakes, so when it vibrates and it goes up and down like that, that's how it goes when it vibrates, right? Because from scala tympani, from this lower part, from this part, the sound, the vibration crosses into the upper part. As it vibrates, a special flap called the tictorial membrane. It's a gelatinous structure that is kind of a flap lying on top of it. So as this part moves up and down, because the flap is connected over here, it's like a hinge, except it's not rigid. It's like gelatinous. But as this thing moves up and down, and this thing is stable, this is stable, relative to the tictorial membrane, the little hair cells are moving left and right. And because they're moving left and right, and their stereocilia are touching, or rather probably are embedded in the tictorial membrane, they get displaced either to the right or to the left, and cells generate changes in voltage, and then they're connected to neurons. So they actually have vesicles here, as a neuron would. And every time they depolarize, they release some neurotransmitter, glutamate, and they are connected to neurons. This is a postsynapse of a neuron. So they are not neurons themselves, but they talk to a neuron in the language that neurons understand, in the language of neurotransmitter, and action potentials are traveling into the brain. Here how it looks like again. Hair cells, stereocilia are touching the tectorial membrane as the basilar membrane comes up and down. Because this thing is fixed, as it comes up like that, the stereocilia are bent in this direction like that. And this is a real picture. These two microphotographs, like the black and white photographs, are pictures made by a scanning electron microscope to show how these things look like. We can see hair cells here, and we can see the stereocilia, so these protrusions that are actually sensitive. And this is a zoomed in version of stereocilia, and they are arranged in this funny way. And these stereocilia are embedded in the tectorial membrane, and these, when displaced, make the cell depolarize and hyperpolarize. And how do they depolarize and hyperpolarize? That's also curious. They depolarize and hyperpolarize because some channels open. And this is wild because these channels use ropes. Like there are for real channels with flaps on them. And these flaps are connected with a rope to a different flap on a different channel. And these channels sit in the membrane. So this is membrane and this is membrane. And if these channels are moved further from each other, the rope gets tightened and the flaps open and ions run through the channels. And these ropes are made of proteins. That's bizarre if you think of that, because usually before we had like a membrane and there would be a channel and the channel was fancy, but at least it was all within one membrane. In this case, it's like mountain climbing of sorts. This channel has a rope that's connected to a different channel that's not even sitting nearby. It's sitting in an entirely different part of the membrane. It's still within the same cell, but it's on a different stereocilia. So if you were to climb this membrane, if you were to become really, really, really tiny, and you would get here and you would start climbing, you would have to go all the way down and then all the way up before you arrive here. So even synthesizing these channels, even placing them is quite a task, right? Because you have to synthesize two channels that are connected by a rope, but then you place one channel on one stereocilia at the base of it, another channel on another stereocilium on the base of it, and then you have to lift them up. So you'll have to move them in synchrony, and you have to be careful because if you lift one too fast, the rope will stretch and everything may break, right? It's pretty hard. And then as these things move back and forth, as they move like that, that's hard. As they move like that, the ropes get tightened and relaxed. So you have to actually place these channels just precisely so that when it sticks like that, it's like semi-open. So tightened but not too tight, so that it would get tightened even more if it moves in one direction. I'm not sure I'll show that. If it moves in one direction, but that would completely relax if it moves in the other direction. It's hard. And then one more thing that I need to mention, in this case, and I'm almost ashamed to say that because I'm afraid that it can be too much for you, but in this case, potassium ions are actually depolarizing the cell when they enter the cell. And this is an aside. If it feels like too much, just ignore it. Just switch off your brain for a few seconds. But if you're like more medical school oriented, you probably want to listen to that because it doesn't affect the grand scheme of things. It's just a really weird physiological and evolutionary story that I don't completely understand myself. But hair cells are exposed to a fluid because they have to be alive, except this fluid is not like normal fluid. It's not like blood plasma. It's not like all other extracellular fluids in our body. It's not full of sodium. It's actually full of potassium, which is totally backwards to everything I ever said in this course before. Every time before, outside of the cell, we had sodium. Inside the cell, we have potassium. In this case, outside of the cell, we have a fluid that is ridiculously rich with potassium, that has more potassium than the inside of the cell. And because of that, when channels open, potassium rushes in. In all other cases, in all normal scenarios, it is sodium that rushes in. In this case, we have more potassium outside than inside, so potassium rushes in. So when these channels open, potassium gets in and depolarizes the cell. Why? Why in this case only everything is not like in any other place. And by why, I mean two different whys. Why it has to be so, in the sense like what's the benefit of having it like that? Why evolution could have created it like that? And how did it even start? Like how do you converge on a system that uses an entirely different fluid that's so extreme? I don't understand that. I don't know that. Probably there is some way to explain that. I don't know it. End of the footnote about potassium. So that's basically the story, but before we finish, I want to try to stress this point once again. So look what happens. We have movements of the air that are funneled by the outer ear into a smaller space, so we have more powerful movement of less air. So the outer ear kind of works like a funnel. Then after that, we have the ossicles that take the movements of this membrane and concentrate it onto a smaller area to make them yet more powerful, so they would be able to move the fluid. And I'm not drawing it to scale, but you get the point. Then we have the movements of the fluid that shake the membrane. And we have to do that because we have to disassemble one sound into all the frequencies that make it. Because we cannot catch up with the sound, it's too fast. We cannot report it to the brain as it is, for the brain to think about it. We have to mechanically disassemble it into frequencies and report frequencies separately. Because once we have detectors along the cochlea, we can report at least whether the frequency is present or absent, right? We'll talk more about it in the next lecture. We have frequency analysis. But then, frequency analysis aside, the movement of the fluid becomes the movement of the membrane. And then because of the way the tectorial membrane works, when the basilar membrane moves, the tectorial membrane that lies on it moves slightly relative to it. Like that. So a relatively big movement of the basilar membrane translates into much smaller movement of the tectorial membrane relative to it. Because it has to be small enough not to become larger than the distance that these hair cells can travel. If the membrane, the basilar membrane moves too much, the tectorial membrane will move too much and it will just break the cells, right? So we have to concentrate all these vibration basically at the cellular scale. We go from the scale of ossicles that are small, but still you can pick them up with your fingers, onto a scale of individual cells by pure mechanical means. But then the way cells detect it is by having actual ropes that are made of individual molecules, proteins, giant molecules, but molecules nevertheless, open other molecules that serve as channels and let the ions through. And it's still the vibration, still the mechanical process that happens here. So these systems of the ear, from the funnel of the outer ear to the ossicles, then the movement of the tectorial membrane relative to the basilar membrane, and then the movement of these stereocilia relative to each other, stretching actual ropes between actual channels and opening them. It's like a ridiculous funnel of mechanical transformations that transform vibrations of the air that you can see with your naked eye, right? If you've ever been near a loudly playing musical instrument, or if you sat in a car with unreasonably loud music and you felt the windows shake, the movement is noticeable, like it can be millimeters at scale, but this movement has to be translated at the movement at the scale of individual molecules. Because ultimately the sound, the movement of the string of a guitar, has to eventually tighten ropes that are made of single molecules. Does it make sense? The difference in scales between what we perceive and how we perceive it is ridiculous. We go from centimeters to not just micrometers, but nanometers if we consider the motion that opens and closes the flap on the channel. And yet this is a purely mechanical transformation. This is bizarre. So remember that. And one last thing that I forgot to mention. Finally, there is another way that makes different parts of the cochlea sensitive to different frequencies. We mentioned two so far, that the width is different and that the thickness and thus the rigidness or flappiness of the membrane is different. It's more rigid closer to the entrance, it's more flappy towards the apex. But there is a third thing that makes the membrane tightly tuned. And this thing is the interaction with hair cells themselves. And that's an interesting story because In 19th century, people kind of figured out that this must be happening when they looked at the cochlea, like why it is there. They would dissect animals, they would dissect dead people, cadavers, and they would find the cochlea and they would be like, how does it work? Probably that's what's happening. But then at some point they actually measured the sensitivity of cochlea to different frequencies by playing sounds to a dead ear, to an ear cut off from a human cadaver. And indeed, different parts of the cochlea were sensitive, but not that sensitive. Like the entire half of the cochlea would get activated. How is analysis of frequencies possible if like half of the cochlea responds to every given frequency? Well, it turns out that dead cochlea are not as nicely tuned as live cochlea, because on top of all these mechanical tuning, live cochlea also have hair cells. And hair cells are sensitive cells, sure, but we have outer hair cells, a lot of those, and inner hair cells that are kind of lonely. If you look at this picture, you would think that it's mostly outer hair cells that respond to the brain, but actually it's the inner hair cells that respond. Outer hair cells are there for a different reason. They shake the cochlea back. When they are shaken at a frequency that matches their preferred frequency, they shake it back. And I won't go into details. There is a funny video in the description that shows a dancing hair cell. You should absolutely watch it. The message here is that while hair cells transmit motion of stereocilia into changes in voltage, they also transduce changes in voltage into motion of the membrane. They work both ways, not just one way. Not only motion causes changes in voltage, but changes in voltage cause motion. And they act as mechanical amplifiers. When cochlea starts to vibrate, if the vibration is close but not exactly the same as that of the hair cells, of the outer hair cells, they don't vibrate back. They kind of move, but that's it. But if it matches their preferred frequency, which is very narrowly tuned, they start vibrating in response, and it creates a sharp peak of sensitivity in this particular part of the basilar membrane, of the organ of Corti. And that's what gives our cochleas in a live human this really nice sensitivity. And then the final step is communicating to the brain. We'll talk a little bit more about it, but essentially each part of the cochlea responds to one frequency, and hair cells spike when it vibrates. So when the frequency is present, this part of the cochlea will vibrate, hair cells will talk to the neurons will send information to the brain. The neurons are sitting in the spiral ganglion. From the name of the ganglion and from the fact that this entire picture is a cross-section, you can figure out that it runs along the entire structure of the cochlea in the same way as all other structures on this picture run. So this is the spiral ganglion, but this is also spiral ganglion, and this is also spiral ganglion. It goes like that. And then it sends information to the brain. So if this frequency is present, there will be spiking here, but not here. Now, if this frequency is absent, but this frequency is present, then these axons will send spikes to the brain, but these axons won't. And thus the brain knows, it would still have to analyze it. Some computations are still required, but at least it will have information theoretically represented in all these fibers that go from the cochlea to the brain, information about what frequencies are present. And if you remember how coding works, we can actually hope to communicate even slightly more. But we'll talk about how the cochlea talks to the brain in the next part. Thank you for tuning in, and see you in the second part.
Time to learn about hearing. Time to learn about hearing.